Go.
Okay, so for number one, big A represents the non-disease allele, and the little a represents the disease allele. So the first one, it gives us the genotypes of a female and a male, and so once you do the Punnett square, you'll end up with 1/2 of big A little a, 1/4 big A, and 1/4 little a. And so since it says that the imprint silences the paternally inherited alleles, so that means that every single one so half of them are going to be diseased and half are going to be normal. And so then with the second one, it gives you the female and the male again, genotypes. And so you'll do the Punnett square again, and you'll end up with, for the genotypes, half are big A little a and half are little a. And so 100 of them, or all of them, will be diseased because of the little a, which is the disease, since it's the paternally inherited alleles are silenced. So then for C, it says that suppose a woman has the triple X syndrome, and so the XICS have been deleted from 2 of the chromosomes, and so how many Barr bodies will be found in each of this woman's cells? So since it doesn't need to delete any, because there's one X chromosome that has it on it, but the rest don't, and so when it goes over it, it looks, and it'll show that it doesn't need to deactivate any, and so there's not going to be any Barr bodies. For D, we just call it the T6 and the X. These activate, and then these ones deactivate. And so it says that the T6 genes on each chromosome, and the cat has 2 X chromosomes, so it says that the T6 on each of those X chromosomes are non-functional, and so the X-is, or whatever, is going to deactivate those on both of those X chromosomes, and so you'll have 2 Barr bodies.
Go.
So for number 2, it asks for an example of a holoandric gene, and we said facial hair growth is a holoandric gene because it's Y-linked genes that are found on the Y chromosome. Then for B, pseudo-autosomal, because it mimics the inheritance pattern of genes found on autosomes, and we use MIC2 as an example of a gene found on both X and Y chromosomes. Then for C, a cross is made between a LGF2 negative male mouse, and same for the female mouse. And what are the phenotypes of the offspring? So we said 2 normal and 2 dwarfs, because maternal is silenced and the paternal is expressed. Then for D, so in sex-influenced pattern of inheritance, an allele can be dominant in one sex while the same allele is recessive in the other. So one sex will display the trait and the other won't. Genes that display sex-influenced inheritance are not found on the same sex chromosomes, they're autosomal. Then for E, genomic imprinting is the epigenetic process that occurs during gamete formation. So it influences the expression of the paternal or maternal alleles of a gene in that offspring. And then dosage compensation is the epigenetic process that occurs during embryogenesis. So that allows females and males to express similar amounts of X-linked genes. Next is F, and so the 2 molecules that form the double-stranded RNA is the X-ist, which coats and inactivates one X chromosome, and then the T6, which produces a functional RNA molecule that is complementary to X-ist. And then last is G, and so the reciprocal crosses that gives identical results, the allele that controls that trait in question are located on the autosomal, and then when the reciprocal crosses give different results, the alleles that control the trait in question are on the X-linked.
Go.
Alright, so for question 3, for part A, we're looking for the genotype of a mom and dad, where the father has OTD, which is an X-linked recessive trait, and the mother is homozygous and unaffected. So, to get across, the dad would have a recessive trait on his X chromosome, and the mom would be homozygous dominant, so she wouldn't have the disease. And then the woman will have a child with a man who doesn't have OTD, and you're looking for the percentage of sons that they'll have with OTD. And so if you cross the mom and the dad here, you'll find that the woman will be homozygous, or heterozygous, and so you'll cross her with a man without OTD, and you get 1/4 for a son that's affected. And for part C, we're looking for the genotype of a male that has a daughter without OTD with the woman. And so the male would be recessive for the trait to be able to get a daughter with the trait. And for part D, all the genotypes and phenotypes found in the cross from part C, you'd get heterozygous female is 1/4, which would be unaffected females. Homozygous recessive is 1/4, which would be affected females. You'd have an affected male is 1/4, and an unaffected male is 1/4 as well.
Okay, so for problem number 4, these are sex-influenced traits, and so this is the goats with beards or no beards. So for the male, it's going to be dominant. For the female, it's going to be recessive, and so that mainly is saying that for like males, it's going to show. So they'll have the bearded trait, and females it will not, even if they have that dominant allele, I guess is how to say it. So for A, it says that their offspring. There are 3, the ratio is 3 bearded to 1 no beard. And this is talking about male offspring. And so what I did is I figured, okay, so if there's one with no beard, I know it's going to be little b, because for the male, that's the only way for them not to have a beard. And so then I thought, okay, with 3 beard, then you're going to have a big B, at least in all of these. But we also know that, and so then I just kind of filled it in from there. And so that has to be little b, and so this is going to be like this. And so for the genotypes and phenotypes, for the male, the genotype is going to be big B, little b, as well as for the female. But the only difference is for their phenotypes. For the male, it's going to be he'll have a beard, and for the female, she'll have no beard because of the sex-influenced trait. So then, for part B, we know that their offspring, it's for the female offspring, and we're going to have 1 beard and 3 no beard. And so what I did for this one is, again, I just put little b in this corner for no beard. And one of these females is going to have a beard, and so we know that that's going to be big B, because that's the only way for a female to have that trait. And then I just filled everything else in. Oh wait, yeah. Because we know that 3 of them have to have no beard. And so it'll be big B, little b, little b again. And so for the genotypes and phenotypes, male's going to be big B, little b, and he'll have a beard. Female will have big B, little b as well, but she'll have no beard. Then for part C, it tells us the phenotypes of the parents, so male with beard and female without beard. And then the ratio is 1 beard to 1 no beard. and then it also tells us that the offspring will be female. And so with this one, for the female, since half of them are going to be bearded, I knew that has to be big B, big B. And so then I just filled in the rest of the chart because half of them cannot be bearded, as well as the male has to be bearded. And so this would be big B, and the female has to be no beard. And so their genotypes, the male would be big B, and the female would be big B, little b, giving her no beard. For D, both of the parents do not have a beard. All the offspring are going to be male, and the ratio is 1 beard to 1 no beard. And so I knew for the male offspring that they at least have to have 1 big B for half of them, and then for half of them to have no beard, we have to have little b's. And so I just filled that in here. We know that one has to be here, 1 big B and 1 big B. And so in order for that to work, then you just fill it out the rest. Little b. And this makes sense because since both parents don't have a beard, we know that the mom will have big B, little b, and then the male has to have little b in order to have no beard.
Go.
Okay, so for number 5, part A, it's asking what happens if you cross a male orange cat to a female tortoiseshell cat, and these big B's and little b's are marking the X-linked trait of coat color. And so if you do the cross, you come out with one orange female cat whose genotype is X with a little b and X with a little b as well. One orange male, that's going to be X with a little b and a Y. One tortoiseshell female, X big B, X little b, and then one black male, which is X big B, and then a Y. Part number B asks how it's possible for a male cat to have a tortoiseshell color, and we learned that tortoiseshells are only possible by the X-linked gene being heterozygous, and so this male would have to have 2 X's as well as a Y, which is why it's not very common. In part number C, it's talking about fruit flies and mutated wings, and so it talks about trying to perform a reciprocal cross to test to see where the trait is found. So we perform a reciprocal cross first with a affected female. So we have these little b's to represent the mutation, crossed with a normal male, and that produces offspring that have the phenotypes of 2 affected males and 2 unaffected females. Because it's an X-linked gene and it's recessive, only the males will show that trait because the females are heterozygous recessive. And then if we switch that test cross and have an affected male and an unaffected female, we'll see that though the phenotype will be 4 of the offspring will be unaffected because again the females are heterozygous for the recessive trait, recessive mutation. And then part D, we're doing this again except we're showing it as an autosomal recessive, so we signified it by little b right here. And so in order for one of the offspring to be affected, it would have to show the genotype of homozygous recessive, whereas all of these are going to show a heterozygous genotype. And so in this one we see that none of them will be affected. And if we perform the test cross the other way, we're going to have the exact same results where all the offspring are heterozygous and therefore none of them are affected.